All the energy for life on Earth comes from the sun. Green plants and certain bacteria use the energy in sunlight to turn molecules of water and carbon dioxide into starch and other compounds. This process is called photosynthesis, and in plants it takes place in the leaves and other green plants. Leaves are green because they contain the green pigment chlorophyll. It's the chlorophyll that turns the light energy of the sun into the chemical energy of starch during photosynthesis. This plant has been growing in the sunlight, so its leaves should have been photosynthesizing and making starch. Now, you probably already know that starch gives a blue-black color when it reacts with iodine. To show that this leaf does contain starch, all the chlorophyll is washed out of it with boiling ethanol. After a few minutes, the leaf is taken out of the ethanol, it's unwrapped, and iodine is dripped onto it. The blue-black color of the leaf shows that it does contain a lot of starch. A leaf that's been kept in the dark for 24 hours, so that it can't carry out photosynthesis, won't stain blue-black with iodine. Now, a solution of chlorophyll like this looks green because it reflects light from the green part of the spectrum. If it's put into a beam of white light, the light from the green part of the spectrum is transmitted, but the light from the red and blue parts of the spectrum is absorbed. But chlorophyll molecules that have been extracted from plants, like the chlorophyll in here, can't trap the energy of light they absorb. They emit it again as light of a different wavelength. A beam of blue light, for example, is absorbed by the free chlorophyll molecules, but then emitted in the red part of the spectrum. This process is called fluorescence. In order to trap the energy of light, the chlorophyll must be in its proper place inside the chloroplast. This is a chloroplast, magnified many times in an electron microscope. It's got an outer membrane, which separates the chloroplast from the cytoplasm, and a lot of internal membranes, which are either free or built into stacks. And it's inside the inner membranes that the chlorophyll molecules are arranged. The chloroplast contains everything that's needed to make starch, not only the chlorophyll, but all the other enzymes and small molecules which are involved in photosynthesis as well. The large starch granule in this chloroplast is evidence that photosynthesis has taken place. It's quite easy to isolate chloroplasts from soft leaves like spinach. The leaves are put into a blender to break up the tissue. Then they're filtered to remove any large unwanted pieces of tissue, and the filtrate is centrifuged. You can keep track of the chloroplasts because of the green color of the chlorophyll inside them, and after centrifuging you'll find that they've all been packed into a pellet at the bottom of the tube. By adding a suitable buffer solution and stirring, the chloroplasts can be resuspended, and when the suspension of chloroplasts is illuminated with the blue light, there's no fluorescence. Unlike the free chlorophyll molecules, chlorophyll held in its place in the chloroplast membrane can trap light, and this light energy can be used in making starch. Starch is a polymer built up from small carbohydrate units. During photosynthesis, what the chloroplasts do is to take carbon dioxide from the air and reduce it, using the hydrogen of water as the reducing agent, to form carbohydrate molecules, which are gradually built up into starch. The remaining part of the water, oxygen, is given off. Now, the process shown here, the reduction of carbon dioxide by water, is the overall process occurring during photosynthesis. The way photosynthesis takes place in the chloroplast is actually much more complicated than this, and takes place in two stages. In the first stage, light is absorbed by the chlorophyll molecules in the chloroplast, and electrons from water are transferred to a coenzyme, so changing it from its oxidized form, known as NADP+, to its reduced form, NADPH. At the same time, ATP is produced. This stage of photosynthesis is called the light reaction, because it requires the direct involvement of light. In the second stage of photosynthesis, the product of the light reaction, the reduced coenzyme NADPH, is used to reduce carbon dioxide to carbohydrate. This process also uses the ATP formed during the light reaction. The second stage of photosynthesis doesn't involve light directly, which is why it's called the dark reaction. But it's important to realize that in plants, this reaction doesn't normally go on in the dark, because the reduced coenzyme produced in the light reaction can't be stored for very long. Production of reducing power during the light reaction can be demonstrated by adding a blue dye to chloroplasts. The reducing power, which normally, remember, is used to reduce the coenzyme NADP, reduces the dye to a colourless form. And this change can be detected because, as it takes place, the chloroplasts return to their green colour. This type of reaction, where the chloroplasts reduce a substance other than the normal coenzyme, is called a Hill reaction. Now, the light reaction of photosynthesis is itself quite a complicated process, because the transfer of electrons from water to the coenzyme NADP requires the collaboration of two photosystems, or assemblies of chlorophyll molecules. Light collected by the chlorophyll molecules in one photosystem causes one special chlorophyll molecule to become excited and expel a negatively charged electron. This electron is transferred to the coenzyme NADP, and the place it occupied in the chlorophyll molecule in photosystem 1 is filled by an electron expelled from a special chlorophyll molecule in the second photosystem when its chlorophylls absorb light. The electrons lost from photosystem 2 are replaced by electrons produced by the splitting of water. The hydrogen ions from water are transferred to the coenzyme NADP to complete the chemical change from the oxidized form, NADP+, to the reduced form, NADPH. The oxygen is given off. And while all this is going on, some of the light energy collected by the chloroplast is also used to produce ATP. Once the coenzyme's been reduced, it can be used in the dark reaction to reduce carbon dioxide to carbohydrate. This process, converting carbon dioxide to carbohydrate, not only needs electrons, it also needs chemical energy in the form of ATP, and the ATP produced during the light reaction is used for this purpose. This equipment measures the rate of photosynthesis by measuring the rate of oxygen production. This is done using an oxygen electrode, which measures the concentration of oxygen in solution. The electrode itself is at the bottom of this chamber, and if the chamber is filled with a suspension of chloroplasts which are photosynthesizing and therefore producing oxygen, the electrode can give a measure of the rate of photosynthesis by recording the increase in the oxygen concentration of the suspension over a period of time. A steep gradient on the chart recorder connected to the electrode shows that there's a rapid production of oxygen. A gradient that's not so steep shows that there's little oxygen being produced. The electrode chamber has been covered with silver paint to exclude all the external light, so that the chloroplasts only receive light of a known wavelength, which enters the suspension through these bundles of optical fibers. The fact that there are two photosystems involved in the light reaction of photosynthesis can be demonstrated because the two photosystems absorb light of different wavelengths. At the moment, the chloroplasts are receiving light with a wavelength of 700 nanometers. Now that's light from the far red region of the spectrum, and because they're green, the chloroplasts can absorb it very well. But they can't use light of this wavelength to oxidize water, so the output of oxygen at this wavelength is very slow. The gradient on the chart isn't very steep at all. But if light of a shorter wavelength is added, oxygen is produced rapidly, and the really important thing is that this production of oxygen is much faster than if the shorter wavelength alone is used. The dramatic drop in photosynthetic activity when the far red light alone is used, that's the light with a wavelength of 700 nanometers, is known as the red drop. What's happening is that the far red light alone excites only one of the two photosystems, the one involved in reducing the coenzyme, and this can't function without the other system. Because of this, the production of oxygen during photosynthesis requires the cooperation of photons of both far red and another wavelength. And the cooperation of both systems gives enough energy not only for the reduction of the coenzyme NADP, but also for the synthesis of ATP. ATP is needed together with the reduced NADP for the dark reaction. So, using the two photosystems together, the chloroplast can produce, in the light reaction, both of the intermediates needed for the dark reaction. The chlorophyll-containing membranes of the chloroplast are not needed for the dark reaction. They've done their work in producing NADPH and ATP. The dark reaction uses soluble enzymes of the chloroplast stroma. But that's another story.